This is the third video in this series, Let's Break Some Sugar. वीडियो में हम बात करने वाले हैं ग्लाइकोलिसिस की. The first pathway involved in breakdown of glucose. ग्लाइकोलिसिस एक ग्रीक भाषा से लिया गया शब्द है जिसमें ग्लाइकस का मतलब होता है शुगर और स्वीट और लाइसिस का मतलब you all know is splitting. This is also known as Embden-Meyerhof-Parnas Pathway, EMP pathway. It is a process in which 1 mole of glucose is partially oxidized into 2 moles of pyruvate in a series of enzyme catalyzed reactions. Glycolysis की एक important बात ये है कि ये aerobic और anaerobic दोनों टाइप के organisms में होता है and it is not oxygen dependent, doesn't require oxygen, और ये माइटोकॉन्ड्रिया में ना होकर साइटोसोल में होता है क्योंकि the enzymes that are required for these reactions or these series of reactions are present in the cytosol. तो आइए बात करते हैं glycolysis की in detail. Glycolysis is the breakdown of glucose and it requires some enzymes present in the cytosol of an aerobic or anaerobic cell. It involves 10 enzyme catalyzed reactions or 10 steps in total, which are divided into 2 sections. First is the preparatory phase, also known as energy investment phase, and the second phase is payoff phase or energy generation phase. The energy that I am talking about is used up and generated as ATPs. So, let's begin. सबसे पहले glucose enter करता है inside a cell. Glucose will enter inside through a glucose transporter, and after it will enter inside the cell must ensure that glucose cannot escape. So उसके escape को prevent करने के लिए glycolysis का पहला step परफॉर्म होता है, that is phosphorylation. और phosphorylation जब भी आपके पास कहीं पे आए, phosphorylation is always associated with an enzyme kinase. Remember that thing. और इस step में जो कि glycolysis का पहला step है called phosphorylation, हेक्सोकाइनेस नाम का एक काइनेस प्रेजेंट होता है जो मैग्नीशियम 2 पॉजिटिव आयन की प्रेजेंस में ग्लूकोज को एक ग्लूकोज 6 फॉस्फेट में बदल देता है. दैट मीन्स फॉस्फेट का एडिशन हुआ है तो वो किसी फॉस्फेट डोनर से हुआ होगा और वो फॉस्फेट डोनर यहाँ पर होता है एडेनोसिन ट्राइफॉस्फेट यानी कि ATP. दिस इज एन इरिवर्सिबल प्रोसेस. रिमेंबर दैट थिंग. After the formation of glucose 6-phosphate, it will further convert it into another intermediate in glycolysis. That intermediate is fructose 6-phosphate. अब glucose और fructose में एक ही फर्क है. Glucose में जो sugar molecule है that is an pyranose, and the glucose and fructose has a sugar molecule which is a furanose. That means a hexagon and a pentagon respectively. यहाँ पर isomerization होता है. The second step of glycolysis is isomerization, which occurs in the presence of phosphoglucoisomerase enzyme. After that, glycolysis का दूसरा irreversible process आता है. This is the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. This step occurs in the presence of an enzyme called PFK1 and magnesium 2 positive ion. PFK stands for phosphofructokinase 1, and also because this is a kinase, यहाँ पे phosphorylation हो रही है. This process is called phosphorylation and it will leads to the formation of fructose 1,6-bisphosphate. After that, fructose 1,6-phosphate will cleave. The next step or the next process occurring here is the cleavage of fructose 1,6-bisphosphate in the presence of enzyme aldolase and zinc 2 positive ion. This is an irreversible process जिसमें fructose 1,6-bisphosphate cleave हो जाता है into glyceraldehyde 3-phosphate, also called G3P, and dihydroxyacetone phosphate, also called DHAP, as shown in this picture. इस ही process में DHAP जो एक molecule है which is a 3-carbon molecule can be isomerized into glyceraldehyde 3-phosphate in a reversible process by the action of an enzyme. This enzyme is triose phosphate isomerase. DHAP के glyceraldehyde 3-phosphate में conversion होने की वजह से we will get 2 molecules of glyceraldehyde 3-phosphate or G3P, which is a 3-carbon compound. और इसी के साथ हमारा पहला फेज यानी कि प्रेपरेटरी फेज और एनर्जी इन्वेस्टमेंट फेज होता है समाप्त. आफ्टर द फर्स्ट फेज एंड्स, ग्लाइकोलिसिस का दूसरा फेज शुरू होता है. दिस फेज इज पे ऑफ फेज और एनर्जी जनरेशन फेज. द फर्स्ट रिएक्शन ऑफ दिस सेकेंड फेज इज ऑक्सीडेशन एंड फॉस्फोरिलेशन कंबाइंड. ऑक्सीडेशन हम अलग अलग तरीके से ऑक्सीडेशन को समझते हैं, but here more relevantly oxidation is the loss of hydrogen from glyceraldehyde 3-phosphate molecule, which will taken up by NAD positive, which converts itself into NADH. Secondly, phosphorylation. Phosphorylation के लिए हमें phosphate group चाहिए जो कि यहाँ पर ATP से नहीं आएगा बल्कि एक inorganic phosphate add होगा into glyceraldehyde 3-phosphate to convert that molecule into a full-fledged stable molecule or an intermediate of glycolysis second phase, which is 1,3-bisphosphoglycerate. All these processes, that means oxidation and phosphorylation, are catalyzed by a specialized enzyme known as glyceraldehyde 3-phosphate dehydrogenase. Moving on to the next step, where 1,3-BPG or 1,3-bisphosphoglycerate is now converted into an 3-phosphoglycerate. Now, 3-phosphoglycerate, by looking at the molecules, you can understand that there is a dephosphorylation here. That means phosphate group is removed, and that is taken up by ATP in the second step of this process, which is substrate level phosphorylation, as shown in this picture. This is an irreversible process. This process is catalyzed by another enzyme, which is phosphoglycerate kinase. Next, the step where 3-phosphoglycerate is now converted into 2-phosphoglycerate, or you can say isomerized into 2-phosphoglycerate by a process called isomerization, catalyzed by an enzyme phosphoglycerate mutase. Now, 2-phosphoglycerate undergo another reaction which is termed as dehydration, catalyzed by an enzyme called enolase in the presence of magnesium 2 positive or manganese 2 positive ions. This dehydration reaction of 2-phosphoglycerate into phosphoenol pyruvate, and 2-phosphoglycerate is converted into phosphoenol pyruvate. And finally, the last irreversible reaction of this pathway, in the last reaction where PEP is converted into pyruvate, this reaction is called substrate level phosphorylation, which is catalyzed by pyruvate kinase in the presence of magnesium 2 positive ion and potassium. Here ATPs are found. और आखिर में we got pyruvate, 2 molecules of it. Something interesting happens here when 1,3-bisphosphate is to be converted into 3-phosphoglycerate in step number 7, catalyzed by phosphoglycerate kinase. In RBCs, this takes a detour, and this detour is named as Rappaport-Lewis shunt. This happens in red blood cells because there is the lack of a particular enzyme in RBCs, which is phosphoglycerate kinase, which is used to convert 1,3-bisphosphoglycerate into 3-phosphoglycerate. That is why in RBCs, because of lack of this enzyme, this pathway take a detour in which 1,3-bisphosphoglycerate is converted into 2,3-bisphosphoglycerate, 2,3-bisphosphoglycerate, and finally into 3-phosphoglycerate. No, there is no direct conversion, and this shunt or this detour is known as Rappaport-Lewis shunt. The remaining 5 steps occur in the second phase. Here, this is reaction number 6. This is step 6, oxidation plus phosphorylation. This is step 7, which is substrate level phosphorylation. Step 8, called isomerization, catalyzed by phosphoglycerate mutase. Step 9, known as dehydration, catalyzed by enolase. And finally, the last reaction, reaction number 10th, which is again a substrate level phosphorylation catalyzed by an enzyme, pyruvate kinase. Now let's summarize the whole pathway. After the entry of glucose inside the cytosol, it is converted into glucose 6-phosphate. The first step of glycolysis, it is an irreversible step catalyzed by hexokinase assisted by magnesium 2 positive ion. This step involves the use of ATP. In second step, glucose 6-phosphate is converted into fructose 6-phosphate, and this step is called isomerization, catalyzed by phosphoglucoisomerase. In step 3, which is another phosphorylation, in step 3, fructose 6-phosphate is converted into fructose 1,6-bisphosphate. This process is catalyzed by an enzyme phosphofructokinase 1 assisted by magnesium 2 positive ion. In this step, ATP is used. Step 4, the fructose 1,6-bisphosphate is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This process is called cleavage, catalyzed by an enzyme called aldolase. Zinc 2 positive ion is present. This process is irreversible. After the cleavage of fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are formed. Dihydroxyacetone phosphate is converted back into glyceraldehyde 3-phosphate in step 5, which is isomerization, catalyzed by an enzyme triose phosphate isomerase. This marks the end of the preparatory phase. After that, the payoff phase starts, where 2 molecules of glyceraldehyde 3-phosphate generated in preparatory phase are converted into 2 molecules of 1,3-bisphosphoglycerate in a step or process which is called oxidation and phosphorylation in the presence of an enzyme glyceraldehyde 3-phosphate dehydrogenase. Oxidation corresponds to the removal of hydrogen, which is taken up by NAD positive, and phosphorylation is done by inorganic phosphate molecule. After that, in step 7, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. This step is known as substrate level phosphorylation, catalyzed by an enzyme phosphoglycerate kinase. This step involves the generation of 2 ATP. After that, in step 8, the 3-phosphoglycerate is converted into 2-phosphoglycerate. This step is known as isomerization, catalyzed by an enzyme called phosphoglycerate mutase. This is step 9, where 2-phosphoglycerate is converted into phosphoenol pyruvate. This step is known as dehydration, characterized by removal of water molecule and catalyzed by an enolase enzyme assisted with magnesium 2 positive or manganese 2 positive ions. Finally, the last step of glycolysis, the step number 10, where phosphoenol pyruvate is converted into pyruvate. This step is the second substrate level phosphorylation, catalyzed by pyruvate kinase and assisted by magnesium 2 positive ion or potassium ions. Finally, we got 2 molecules of pyruvate at the end of this whole pathway. One last thing that remains to summarize is Rappaport-Lewis shunt, present or known to operate in RBCs. Remember that 7th step in glycolysis, the first substrate level phosphorylation, where 1,3-bisphosphoglycerate's conversion into 3-phosphoglycerate is catalyzed by phosphoglycerate kinase. This is the direct conversion. But in RBCs, it takes a detour via 2,3-bisphosphoglycerate. That means there is no direct conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate, but it takes a detour via 2,3-bisphosphoglycerate because of the lack of an enzyme, which is phosphoglycerate kinase in the RBCs. Now, as an overview, in glycolysis, there are 4 ATPs formed, out of which 2 ATPs are used up in preparatory phase and 2 ATPs are the net production. And besides 2 ATPs, there are 2 NADH also formed in this pathway. So that is all about this whole pathway. I will post an infograph of overview of glycolysis where you can see the reactions where ATP used and ATP are generated, and also where NADH is formed. Here our video ends. See you in the next video. Thanks for watching. Remember, this is the mnemonic.